All right, so evaluating predictive performance and setting decision thresholds. We build a model. Before we apply it, we need to have some sort of test, some sort of measure of whether we have some predictive performance there that's acceptable for our application. So that's what we're going to focus on. AUC curves, different approaches to validating models. There's a whole lot of debate in this field as to which are the best approaches, and I'll touch on some of those. And there's a whole load of different tests that you can do, different ways of doing it that you will see in the literature. What I'm going to try and do in the next hour or so is to get everyone up to speed with what the basics are, what the main approaches are that you will see in the literature. I'd like to think over the next 5 or 10 years we'll be moving away from some of these methods towards some better approaches, but the goal for now is to really, emphasize where we're at and what the main tests that you will see and the main tests that you'll get out as kind of defaults from tools like Maxent or Biomod or other approaches in R. Okay, and part of what we'll do is discuss setting decision thresholds, which we mentioned a little bit yesterday. I'll talk you through that. It's a bit of an aside in some ways, but it fits very nicely in this section because it's really crucial to understand how you set decision thresholds if you are going to understand how some of these validation statistics work. Okay, so hopefully that will become clear, but that's why it's kind of sandwiched in there, and we'll take a bit of a detour to talk about those. So let's revisit where we were up to. This is just sticking with our flow diagram that we're using as a kind of structure of the things we're going through. Of course, we've talked about getting hold of distribution data, presences, absences, collating your database of environmental data, pre-processing potentially to get some predictive variables that have some biological meaning for the kind of questions that you're addressing and the organisms you're dealing with. We then talked about the algorithm, the actual model that you're going to apply, Maxent or artificial neural networks or generalized linear models. We talked a little bit about, for each of those models, you're going to try and calibrate the model or parameterize it as best you can. And then we talked about mapping that prediction back from environmental space into geographic space. So we plot a map. We plot a map that shows the localities of environments that are similar to where you've already observed the species. Okay? And we're going to focus now on, is that prediction any good? Let's evaluate the predictive performance. So first thing we need is some data, right, that we can test the performance against. We need some sort of data set that we can use to ask, well, is the model any good? And there are various ways of doing that, and I'm just going to kind of schematically illustrate those. Suppose this is all of our available data, all right, 100% of it. It might be 20 records for some of the examples that I have shown and will show for Madagascar. It might be 1,000 records for some of the plants we have in Europe. It might be more than that for some records from GBIF that you've downloaded. So regardless of the number of localities, let's just represent this as kind of all of our data set. Then what we're going to do, of course, is build our model on this some set of data, and then we're going to make our projections that we apply. This is kind of the interesting application. We might project into the same region. If you're doing reserve planning or something, into a different region, maybe you're interested in invasive species, different time slice for climate change, and there's some work projecting across different spatial resolutions and things. But that's kind of the end goal. That's the application. And we'll come across some terminology then. So calibration is, in effect, like building your model. Another word that's often, in effect, a synonym, but comes particularly from the machine learning literature, would be training. You train a model. You train a model to learn a pattern. So calibration and training are the data sets that you would use to actually build the model, to construct the model in niche space. And then we're going to evaluate the model. Another term that you might use is test. You'll see them in the literature. So kind of calibration and is building the data set, and then evaluation is evaluating how good it is, or training and testing. So one thing that you can do is take all of your data, calibrate your model, and then evaluate against the same data set. What's the obvious limitation there? Overfitting, precisely. Exactly what we talked about yesterday. You can very strongly fit to your data set, but then when you present the model with some new data that it hasn't seen before, the model can perform very poorly on some new data set. So one of the risks with this approach is overfitting, and that's classic across all kind of statistical modeling type or machine learning approaches. We want to avoid overfitting. You'll still see that published in the literature, particularly in some of the earlier literature, but it's not really the way to go. I strongly discourage you from that approach. So what's another approach? Well, this is kind of, in some ways, a kind of gold standard. This would be ideal, that we would build our model based on all of the data that we have available. We would calibrate the model, we would train the model, and then we would evaluate it specifically for our evaluation. Okay? So one example that, or a couple of examples of this kind of approach that you'll see later on today would be the work that I'm going to present in Madagascar, where we actually send field crews out to collect new data. So we calibrate on all the data that we have, we build the model, we project it to the same region, back to Madagascar, and then we send field crews out to collect new data that we use to test whether the model's performing well. Another example that you'll see is South African invasive species that we'll talk about a little bit later, where those authors collected, they built their models for South Africa, so they calibrated for all the data they had for South Africa, then they projected the models to the rest of the world, and for example in California or in Australia they got invasive records from localities where the species was invading in the different regions, and then used those points to test how good the model was. Okay, so you calibrate on all the data that you have, and then you collect some new data that's specific to your application. That's all very well, that's all very good. Often we can't do that, and really the most common approach that's often also combined with that kind of gold standard of what you would ideally do is to do some sort of data splitting, some sort of data partitioning approach. So here we divide our 100% of the data that we have into two parts, one of which we're going to use for calibration or training, and the other we're going to use for testing or evaluating. And a common split would be something like 70% for training and or for calibration and 30% for test. But you might use 60 and 40, or you might use 80 and 20. There's not really a golden rule, but 70-30 is a kind of sensible split. Okay, so you calibrate on the 70%, and then you ask, you generate some of this kind of statistics that we're going to talk about. You ask how well does the model perform on the remaining 30%. Then what would be usual, that you would get some validation statistics based on your 30%, and you would then use put all your data back together and use 100% to actually make your projection. Usually you don't want to actually be leaving data points out for your actual application. So you'd do a data split, test how well the model's performing, then put all your data back together and make your actual projection. Okay? One step better than that you like, if you like, and that's really the standard way of doing these things in the literature, is to do what's called basically usually a k-fold partitioning or a cross-validation type approach. So in this one, again, this is our full data set, might be say n observations. And what we're going to do is, instead of just doing a kind of one split into say 70 and 30, we're going to split into say, if it was, to get the terminology, if you were doing 5-fold partitioning, you would split into 5 folds, 5 different parts. Obviously in this case you'd have 20% of the data in each of those parts, and then you're going to build a model based on 4 of them and test against 1 part. So you've got 100 points, you build the model based on 80 points and you test against 20 points. And then you repeat that, of course, leaving one of your partitions out each time. So with k-fold partitioning you will get 5 statistical results once for each time you leave a partition out. And then you would usually present, say, the mean of those results across the 5 folds, or the range, or often both, the mean and the range of the results that you get. And And of course, you could do that with any number of k-folds, usually between 5 and 10. The more data that you have, often you might decide to split into more parts, but you want to make sure that that part that you're leaving out has enough data in it to actually test the model. Okay, but usually you'd be working with 5-fold or up to 10-fold partitioning. Not really a golden rule. Just one thing to add. We're not gonna have a session on this, but I'd be happy to discuss it if people are interested. We developed an approach if you have a very low number of records. If you have, say, like some of our cases from Madagascar, we have only, say, 15 records or up to 20 records for some of these organisms, which is very, very few records to actually validate the model. So we generated a test. It's a kind of jackknifing approach. It's published in a paper in Journal of Biogeography in 2007. As I say, I'm not going to have a session on that in this kind of couple of days, but I'm happy to talk about it. And those of you who are interested in reading that paper, in effect, what we talk about in that paper is a jackknifing approach where we set k equal to n, where n is the number of points that we have. So what we actually do is each partition is just a single point. So if we've got 15 points, we run 15 models, each time leaving 1 point out, and we test how well we can predict that 1 point based on the other 14. Conceptually, that's fairly straightforward. It raises some difficulties. We can't apply the usual kind of test statistics for various reasons, but we developed an approach where you use a different kind of test statistic, and that's written up in a paper in 2007. But I just wanted to flag, if you're interested in low sample sizes, there's that approach out there. In effect, all it is a twist on this idea of k-fold partitioning, but k equals n, where n is the number of points. Okay? Now let's take our little aside, because we're going to want to understand thresholding for various reasons, but one of them is to understand how these test statistics work. So in effect, when we talk about setting a threshold, some people will refer to it as like a decision threshold or something like that. We're basically just saying, if our model, which most of the ones that we work with are, if our model outputs a probability of occurrence or a suitability score in some cases, a continuous value, say, ranging between 0 and 1, can we set some threshold, some value above which we're going to assume that the species is present, or at least that the habitat is, the environment is suitable? So we're going to go from a kind of probability of occurrence, that you're seeing there is the species is the marbled salamander in North America, straightforward Maxent, default Maxent output, a logistic output from Maxent. That's a probability of occurrence, right, ranging from 0 to 1, going from the cold blue colours to the hot red colours. So that's very well, but sometimes we want actually this kind of binary prediction where we set some threshold above which we say, well, above, say, 0.6 or 0.7 or 0.3, whatever the value may be, we're going to classify that as suitable, and anything under that we're going to class as unsuitable. So we go from a continuous prediction to a binary prediction. Okay? And there are tons of ways of doing this. A couple of papers that I should actually have on this reference list, I should add them, particularly by Kanran Liu, published a couple of papers over the last few years looking at reviewing and testing various ways to set decision thresholds. He published one paper that was focused on when you have presence and absence records, and another paper that was focused on when you have presence only data. So there are tons of methods out there. Kanran Liu et al., L-I-U, would be a key author for if you really want to dig into some of the details here. But what I'm flagging here are just some of the two simplest, most straightforward and most commonly used approaches. Very straightforward. So same model that we saw on the previous slide, but thresholded in different ways. All we've done here is said, in the version on my right, your left, that's basically saying we're going to use a 0% emission rate. We're going to start talking about things like emission rate. Essentially, that's just saying we're going to set a threshold so that none of our occurrence records are left out. Okay? And I should emphasize that we're doing this based on the calibration data or the training data. We're not doing this on the test data. This is all part of the process of building our model. And we set the threshold. If we set the threshold on the test data, then we're just going to fix that we do really well on the test data. You know, that's not a good test of the model. So we do this on the calibration data. So if these were all the points that we used, the black stars that we used to build that model, we've just set a threshold which says we're not going to exclude any of them. We're going to shrink the distribution as small as we can until we hit the point where we're going to start excluding records. Okay? So in practice, that means that if all our records, obviously, all our calibration points have a prediction associated with them, a value, you would hope that those are relatively high predictions, right? You would hope that where you have species presences, that the model is predicting a high probability that the species should occur there. Okay? So all you're going to do is basically look at all those probabilities that are associated with a presence record and choose the lowest one. So if you've got 10 records and their values of, you know, predictions of 0.9, 0.95, 0.8, 0.87, etc., and then there's one of them that's 0.7, then we have to set the threshold at 0.7 or just below 0.7 so that we ensure that we include all of our records in the prediction. So we're setting zero omission. Another approach would be to say, well, let's actually allow some of our points to be omitted from the prediction. So we might say, in this case, we've got a 10% omission rate on your right as you're looking at it. So that's setting a threshold where we shrink the distribution down, until we are leaving out 10% of our records. So suppose you have, we talked yesterday about different types of data. Suppose you've downloaded a bunch of data from GBIF and you've got 1,000 records. But we talked about all the kind of uncertainties. Maybe there's some errors in there with the georeferencing, with the species IDs, etc. So you might actually be willing to leave out, say, 5 or 10% of your points to shrink it down a little bit more to the actual core points. So you're predicting 80 or 90% of the points, but you're accepting that you're going to leave a few out. Okay? So that would be this approach. You might select in that kind of instance that you're going to allow some of the points to be left out. Now, that's particularly going to be the case if you've got kind of ecological outliers. By which I mean, if you've got just, say you've got 100 points and you've got one of them that's in a completely different environment because maybe it's ecologically interesting that it's in a different environment, but maybe it's an error, maybe the ID's wrong, something like that. That one point is going to massively impact your distribution if you use a 0% omission, because you're going to have to include it. So all your records, 99 of them might be predicted at 0.9 probability and above, but one of them's 0.3, so you're going to have to set your threshold at 0.3, which will vastly impact your prediction. So another thing to bear in mind, what's going to happen to your prediction as you increase the threshold, as you increase it from 0.4 to 0.5 to 0.6? What's going to happen to your prediction? Is it get smaller or get bigger? The threshold goes up, the prediction is going to get smaller, right? If you think about, if you set the threshold at 0.1, then anything that's predicted above 0.1 is going to be classed as suitable or present. So you're going to get a big area. Once you start getting up to 0.9, you're shrinking down to a smaller area. Okay? So you increase the threshold, you reduce the area that you're predicting as present. So in this case, we're saying, well, let's reduce the threshold to what might be called like the lowest presence threshold is a term that's used. But to set it for zero omission, so you're using a lower threshold, which gives you a large predicted area to make sure you include all your points. As you increase the threshold, the area shrinks and you omit more points, so you would be allowing some to fall outside your prediction. So your omission rate, the rate or the number, the proportion of points that you have, occurrences that are left out, the omission rate goes up. So that's two common ways to select a threshold of occurrence. One thing to just ask, why would you actually want to set a threshold of occurrence? One thing to bear in mind is you are in effect, you're dumping data. you're ditching data, right? Because you had this continuous data set and you're just chopping it into a binary data set. So why would you want to do that? Well, be careful, because often you don't, because as I say, you're removing some of the complexity, you're removing some of the information that you have. But sometimes it is useful. Sometimes you want to summarize model output based on some sort of quantitative criteria. So often, you know, I've done this, I've presented these kinds of results to policymakers and that, and they, as we talked about yesterday, you know, they want a binary prediction. They don't want this fuzzy probability of occurrence. Just, you know, tell me yes or no, in effect. Setting a threshold in this way based on some objective quantitative criteria of, you know, say you're going to set zero emission rate is defendable, is at least a quantitative, robust way of actually transparently saying, you know, we're going to convert to a binary prediction. So there might be some policy implications, or there might be some reason why you actually just want to have a yes or no answer. Often binary predictions are actually kind of useful in different analyses, like I've listed here, multi-species reserve planning or estimates of species richness, where you're actually trying to combine models from lots of species. So you're not just interested in one, but you're trying to kind of pile a lot of species on top of each other, and then dealing with probabilities for lots of different species becomes tricky. Doable, but tricky. And sometimes it's preferable, you know, if you want an estimate, for example, of species richness, for each species you want a binary prediction. Well, is it likely to be present or not? So there are some applications where you really will want to set a threshold, but don't think, oh, I must set a threshold. It's only you want to have a reason for setting a threshold if you're going to do so. So there are some applications, but also for us it's just important to understand this way of setting binary predictions in order to understand how the validation statistics, how some of those statistics are generated. Okay, so it's going to be important as we flip back now from our little detour using calibration or training data to set thresholds. Let's flip back now to asking what do we do to actually test the performance of the model once we've, you know, actually built it based on some training or calibration data. So this is going back to some of these conceptual diagrams again, just one of them for now, but trying to illustrate the types of results that you would expect to get or that are possible when you're evaluating a distribution model. Okay? So again, conceptually, we're in geographic space here, x and y coordinates. Again, we've got our actual distribution or our actual occupied area shown in the grey shading, similar to the diagrams, the potato diagrams from yesterday. And then we've built a model and we've got these kinds of predictions. Remember, we referred to kind of type 1, type 2 and type 3 predictions, kinds of results that you would expect to get theoretically from these models. And then what the little red symbols are on top of that are assuming that this is our test data. All right, these are our test data points that we've been out and collected them after collecting the model, after building the models, or we've just done some sort of k-fold partitioning, you know, partitioned our data. We use 70% to build the model, and then this is our 30%. But these are our test points, right? So when we plot those onto the landscape, let's ask, you know, we go out and visit each of those points, and we're going to look at the kind of predictions or the kind of test results that we would expect. So first, we're going to refer to true positives, the little x's. They're true positives because we predicted that the species would be present there. We went, we found that it was. So it's a true positive prediction. All right? It's on the actual distribution and it's within our prediction. So that's good, right? That's undoubtedly a good result. Another type would be a false positive prediction, the little squares. So we predicted that the species should be there, we went there, and we found that it wasn't there. It's a false positive prediction. The little circles, false negative predictions. We predicted that the species wouldn't be there because it's outside our distribution model. We went and we found that it was. It was actually part of the actual distribution. So that would be a false negative prediction. Not a good prediction, right? And then a true negative prediction, a good prediction, is that we predicted that the species wouldn't be present, the little minus signs, and it wasn't present. Okay? So those are the 4 results that we can expect to get. This is, of course, assuming that we've got a binary prediction, which is why we've just learnt about setting thresholds. Okay? So we've got a binary prediction. There are 4 results that we can expect. True positives, false positives, false negatives, and true negatives. That's kind of what we have to work with, and that's assuming that we're collecting presence data and absence data. So that's what we have to work with to generate some statistics. And as I say, we're only going to look at a couple of the main ones, but in effect, most of the statistical approaches are in some way based on those 4 possibilities. And we put them in what's often referred to as a presence-absence confusion matrix, where we're saying this is what's recorded present, this is what's recorded absent, and then predicted present, predicted absent. We're just going to give them letters A, B, C, and D, true positive, false positive, etc. Okay? It's the same information, but you'll often see that in papers or in textbooks in terms of how we're going to evaluate the models based on this confusing matrix. But that's all it is. If you think back to the previous slide, it's just those 4 possible results. Something I referred to yesterday, but I'm going to flag while we've got this particular diagram up. Remember this type of prediction. We referred to it yesterday as a type 3 prediction. This is if you're interested, say, in an invasive species application or a reintroduction or something. We're predicting that the species could be there, but it isn't actually there. Okay? Now, if you're evaluating your model with negative predictions, so you go out to that site and you find that the species isn't there, so it's a false positive prediction. If you use a test statistic that punishes your model, if you like, it says your model isn't very good because it's making that kind of prediction, then there is an argument that that's really quite fundamentally problematic, right? We want to be very careful when we're using absence data. If we're interested in this kind of prediction, which theoretically from yesterday morning we should be, that's a kind of prediction that we would expect the model to make, and it's, you know, it's a good prediction, but we don't want to punish our models for making false positive predictions because, you know, that's a good result. So remember when we talked about the model comparison yesterday, which model's best and how do we evaluate model performance, and some of the analyses would include presences and absences. That was my kind of caveat, that that's all very well, but just be careful because you're punishing models that are making good predictions just because the species isn't actually found there, but they might actually be the kind of predictions that we want to make. Okay, so I'm just going to flag that for now. And that's also one of the reasons why we often want to evaluate models based only on presence records. Of course, often we only have presence records. Suppose we're using museum data, we only have presences, we do some sort of data partitioning, 70-30 or k-fold partitioning, you know, we only have presence data. So in practice, we often only have presence data, but I would strongly argue that in theory, we usually only actually want presence data anyway to evaluate the models. So what can we do with just presence data to start with? We will talk about presence absence in a minute. What's the most simple thing that we can do based on our set of results, but only including where you're recording presences, so only including presence test points. What's the most basic thing that we can do as some sort of simple test statistic? Well, we can measure the proportion of the observed presences that were correctly predicted, right? It's just A over A plus C. The true positives over, you know, how many you got right out of the total number of presences that there were. Did you get 9 out of 10 right, 5 out of 10, 40 out of 100, whatever. So it's just a proportion of observed presences that are correctly predicted. Some of the terminology here, that's clearly a simple measure, but you'll hear it referred to in the literature as sensitivity is a term, or the true positive fraction. They're all just this, what I would term in simple language, it's the proportion of the observed presences that you're getting right. Okay? So here's an example of that actually applied in practice. You're going to see a few examples from Madagascar because it's one of my key interests. This is our somewhat evil leaf-tailed gecko, Uroplatus sakurae, and so it's the same species, the same set of occurrence records, but with two different partitions. Okay, so the black data were those that were used to calibrate the model, and the red data were those that were used to test the model. And of course, we could have many more partitions and things, but I'm just showing you two different partitions of the same data. These happen to be maxent models, but that's neither here nor there for our discussions right now. We built the model, we set a threshold of occurrence, so you're only seeing the grey areas there are those areas that are predicted to be suitable, high probability of occurrence. We've set the threshold based on the black points on the calibration records. In this case, we set the threshold with 0% omission, so you won't see any black records that are omitted, hopefully, from the prediction, because we've forced it that all the calibration points are included. There we go. So then what we can do is ask, well, how many of our red points are we correctly predicting? Right? So there happens to be 7 test points. So in one case, our success rate, you see it down the bottom, was 4 out of 7. And in the other case, it was 6 out of 7. How many of those red points, how many of the test points fell on the prediction? If you think about it more conceptually, how many of them had a higher probability of occurrence than the lowest predicted calibration point? Okay? So, that's all very well. There's a test statistic based on only presence data. The problem here is that I can be 100% correct every single time. My success rate, because I'm brilliant, could be 100% every time. Because all I would do is just say, well, I'll just say that the whole of the study area, I'll predict it as present, and therefore I will always get all of my presences correctly predicted. So there has to be another part to the story here where we're going to punish models that predict too large an area as present. Okay? Otherwise, in effect, I'm not brilliant, I'll just cheat. All right? So what we actually do in reality is pair the success rate with a statistical test of significance to ask, well, is that success rate statistically significant based on the proportion of the area that we're predicting present? So we would find if we're predicting 100% the area as present, well, that's all very well, but the model isn't any use because it's not statistically significant. So how do we do that? Well, the common way of doing it is to use a binomial test. And I'll quickly talk you through a binomial test. I'm sure a lot of you will have come across them in other kind of statistical classes. I won't go through it in too much detail. But in effect, what we're going to do is use the proportion of the study area that we are predicting as present as our kind of null hypothesis of the likelihood of being successful at random. So if we predict all of the study area as suitable, then our likelihood of being successful based entirely at random is 1. Right? We're always going to be present. We're always going to be predicted correctly. If we're predicting half of the study area as present, then our likelihood of being correct entirely at random is 0.5. If we predict only 0.1 of the study area as being present, then our likelihood of being successful at random is 0.1. Right? So we'll come back to it, but just quickly then an overview of the binomial test. I just pinched this from Wikipedia because it was as good an explanation as anywhere. Just quickly, suppose that we're working with a die that attaches importance. We're working with a game where getting a result of 6 when you roll the die is, what you're after. That's the winning hand, if you like. And we're playing this particular game, and we roll a 6 51 times out of 235 throws. So the success rate is 51 out of 235. Compare that to our success rate in our Madagascar example was, 6 out of 7 or 4 out of 7. Okay, so that's that comparison. So what we're asking, is that rate of success significantly higher than would be expected purely by chance? That's in effect what the binomial test is going to test. So we're really testing this null hypothesis. Have we got a fair die, or is it loaded? So how do we do that? We use this binomial test where the number of trials is 235. That's the number of rolls we had, or in our case, the number of test points. The number of successes was 51. In this case, it was the number of sixes we got. In our case, it was the number of the test points that we predicted correctly. And the probability of success in each trial was 1 out of 6. Now, our probability of success in each trial is the proportion of the study area that we're predicting present. As I said, if you're predicting 70% of the study area or 0.7, a proportion of 0.7 of the study area as present, then your probability of being successful entirely at random, if you just randomly throw a dart at Madagascar, your probability of being successful is 0.7. And then the way the binomial test works, and there are online calculators, or R will calculate it in a flash, you just add up the probability of getting exactly 51 sixes, 52 sixes, 53 sixes, all the way up to 235 sixes. If you work it out in this case, the probability of getting 51 or more sixes on a fair die is 0.027. So, of course, in biology, we tend to work with a significance level of 5%. So we would be saying that in this case, because that value is below 5%, it's below 0.05, it's pretty unlikely that that set of results came from a die that is not loaded. Okay? So that, in effect, is just the binomial test. So let's just take that back to our presence-only evaluation data set for Madagascar. What we would actually present is the success rate. We would calculate the proportion of the study area that is predicted present. In this case, it was 0.231. So about a quarter of Madagascar, a quarter of the landmass is grey, is above our threshold, is predicted as suitable. And that gave a binomial probability based on that success rate and that null hypothesis of what the probability of being successful at random is, gives a binomial probability of 0.05. So a little bit above significance, not significant. In this case, there was a higher proportion of the study area that was predicted present, but in this case we got 6 out of 7 right, and the result came out as significant. So when you often see in papers presence-only test statistics pairing a success rate with a binomial test of statistical significance, that is in effect what they're doing. And there are limitations of that are widely discussed, and there are efforts being made to come up with improved approaches, better ways of doing it, but that's a very commonly a very common approach that you will see in the literature. It's a success rate combined with a test of statistical significance, which is dependent on checking that you're not cheating, in effect, by making sure that the proportion of the study area that you're predicting present is not so large that the model is not necessarily wrong, but isn't really very useful. Because it might be that for a particular species, the whole of Madagascar is suitable. So it's not necessarily wrong, but it's not really telling you anything that's statistically significant. Let's now think about presence-absence test statistics. So now we're actually not just going to use where we've got recorded present, but we're also going to use where we have recorded absence. So this is where we've actually gone out in the field and we've collected those absence records that, as we know, have caveats associated with them. So we've talked about how from our confusion matrix we can generate the proportion of observed presences that are correctly predicted. Of course, we can do exactly the same with the absences. We can work out the proportion of the observed absences that we are correctly predicting. And you'll often hear that referred to as specificity. So in the literature, you'll often hear people refer to how well you're doing with the presences in terms of the measure of sensitivity, and how well you're doing with absences in terms of a measure that they refer to as specificity. Okay, but it's simple. It's the proportion of the observed absences that you're correctly predicting. So this brings us to ROCs and AUCs. Who's kind of run a distribution model and got an AUC score out? Right. So we're going to try and unwrap what exactly that score is, and it's based on what's called a receiver operating characteristic curve. So this is an approach that uses presences and absences, so the test data of presences and absences, and that isn't dependent on setting a threshold. At least your actual final statistic that you get out isn't based on setting a threshold. So it's not that you have to select a particular threshold and then have a result at that threshold. What this is going to do is test across all the possible thresholds that you could have selected. So you need to understand how to set a threshold in order to understand how to generate the statistic, but the result, as you'll see, is across all the possible thresholds. So it's good because it's often referred to as threshold independent. It's a test that doesn't matter on your selection of, whatever threshold you want to choose. So let's start here with, I'm going to try and break this down to simplify what this receiver operating characteristic, or ROC curve is. And then we're going to talk about the AUC, is the area under the curve. It's really the area under the receiver operating characteristic curve. All right? So let's just focus on these two plots here, what we're trying to present here. Similar kinds of things that we've been looking at yesterday. What we've got is some sort of frequency, so the frequency at which you get that result, and then the predicted probability of occurrence. So this is our, say, a maxent output or any other different model that's predicting a suitability or a probability of occurrence ranging from 0 to 1. And then we've got two sets of data. We've got our set of presences and our set of absences. So these are our test points. We've got some test points that are presences, some that are absences. What we would hope is that the set of presences have a frequency plot where they're generally at higher probabilities, right? You would hope that usually your presences you are predicting with a higher probability of occurrence. So that's what we're trying to depict here. This is our set of presences, and this is our set of absences. So the absences tend to have lower probabilities of occurrence, and the presences tend to have higher predicted probabilities of occurrence. That's what you'd hope, right? And a better model is going to look like this, and a less good model is going to look like this, because the set of absences and the set of presences overlap more. So there are more cases where you have probabilities of occurrence where you're getting both presences and absences. So a perfect model would be able to distinguish those two frequency plots exactly. You'd be able to draw a line here where at a certain threshold you precisely divide your presences from your absences. Okay, so that's what those plots are trying to distinguish, or to illustrate. So what is the ROC plot? That's on the left as you're looking at it. This is kind of by convention how it's plotted, and you'll see hopefully why. What we're going to plot is the sensitivity against 1 minus the specificity. Okay, so that's the terminology that you will tend to see, but let's break it down into some more simple language. What we're going to plot really, so the sensitivity, remember, is the proportion of the presences that are correctly predicted. And then, partly because of convention and partly because you'll see it just works out nicely in terms of how we draw this plot, we're going to plot that against 1 minus the specificity, which is, of course, the proportion of the absences. Well, the specificity is the proportion of absences that are correctly predicted, so 1 minus is the proportion of the absences that are incorrectly predicted. Okay? So we're going to calculate each of these values, the proportion of the presences that are correctly predicted and the proportion of the absences that are incorrectly predicted, across all the possible thresholds that we could. And then we're going to plot them on this graph to make a curve, an ROC curve. All right, so let's break that down. Let's start here. Let's first of all start with a threshold of 0. Okay, so if we set our threshold to start with at 0, that means that we're predicting all of the study area as suitable, right? Anything above 0, we're going to give a habitat suitability of 1. Okay, so in effect, the whole of the study area is predicted as suitable. So how are we doing? Well, we are predicting all of our absences incorrectly, because we're predicting all of our absences should be presences. And how are we doing on our presences? Well, we're predicting all of those exactly right. Remember, we just went through that with the binomial test. If we predict all of the study area as present, then we're going to get all of our presences right. So what we're doing is, here's our first point. The proportion of the presences that are correctly predicted is 1, and the proportion of the absences that are incorrectly predicted is also 1. Okay? So we're going to start top right on our plot. Now that's what's going to happen as we move this way along this axis. Okay? We're going to start moving this way along our curve. So our proportion of absences that are incorrectly predicted is going to start dropping off, while our proportion of our set of presences that are correctly predicted is going to stay high. Okay? So we're going to start coming along here as we increase the threshold. Now, if we had the perfect model, we would expect to reach the point, if these two frequency plots are totally distinguished from each other, as I said, we would expect to reach a point where we could distinguish all of the presences from all of the absences. So if it was a perfect model, we would expect to reach the point where we're here. We're getting all of our presences correctly predicted, and we're getting all of our absences correctly predicted, or the proportion of the absences that are incorrectly predicted is 0. It's a bit of a mind twist, right, to get around how this plot works, but you'll get used to working with them. Okay, so a perfect model is going to come along here, and then it's going to come down here. It's going to follow it's going to go along the top and then down the y-axis as we move our threshold from 0 towards 1. Now our less good model, think about it as we increase from 0 to 1, so the blue plots here, we're going to start dropping off the proportion of presences that are correctly predicted earlier, if you like. Remember, because there's this more overlap here. So we're going to have more area where we're confusing our test presences and absences. So that curve is going to drop off a lot quicker. All right? If these two plots, these two frequency plots, were entirely on top of each other, then in effect we'd have no way to distinguish between the two of them, and as we move from 0 to 1 in terms of changing our threshold, we would just be plotting this 1-to-1 line, because you'd be decreasing the number of presences and the number of absences that you're getting correct just at random, basically. You'd have no way of distinguishing between presences and absences. So the bottom line in practice for how to interpret this is a better model is going to have a curve that fits higher along the top and then drops down the y-axis. The ROC curve is going to plot more towards, it's going to be pushed up towards the top left of that plot as we're looking at it. A less good model is going to move towards the 1-to-1 line. So remember, this was a better model than this. You can distinguish the presences and the absences easier. So it's going to plot as the red line there, closer to that top and then the y-axis. And a totally, in effect, useless model that has no way of distinguishing presences from absences, because those two frequency plots sit on top of each other, is going to just follow that 1-to-1 line. So with that in mind, with that way of thinking about the models and the data in mind, what we do is calculate the area under this curve. Because the area under is not going to just tell us about a particular point at a particular threshold. It's going to say across all of the thresholds, how well's the model doing. So a perfect model is going to follow along here and down here, remember, along the top and down the y-axis. So the area under that curve, it's really not a curve, it's just a two sides of a square, is going to be 1. It's going to be 1 unit. Whereas your totally random model that has no way of distinguishing presences from absences is going to have a value of 0.5. The area under the 1-to-1 line is 0.5. So your AUC curves will range, or your AUC values, area under the curve, will range from 0.5 from a totally random model that has no predictive ability to 1 for a perfect model. Okay? So that's what your AUC curves are showing you. Sorry, your ROC curves and your AUC value, your area under that curve is telling you. So a good model is going to have a higher value, closer to 1, and a poorer model is going to have a value closer to 0.5. So what's a good result? Very, very subjective. And again, there's a lot of debate as to how best to evaluate models, and I think it's one of the areas in this field that still needs a lot more attention, particularly to think about how we use absences, or really whether we should be using absence data at all. But at least to help you get up to speed with what's in the literature and understand the kind of current state of affairs, if you like, some very subjective guidelines for interpreting AUC curves. This is from a very well-known paper that's not specific to our field at all, but is saying if you've got a value from 0.5 to 0.7, you've got very poor discrimination. Pretty good, reasonable, 0.7 to 0.9. Anything above 0.9, you've got very good discrimination. And that's kind of what's largely used in our field, that if you get AUC curves above 0.9, you're doing pretty well. I think we're finding in practice that it's a bit of a low bar. It's kind of pretty easy to do in many cases. So we need to think about this some more. One thing as well that I will emphasize before I kind of open it up to questions is that some of you who will have worked with tools like Maxent and other methods, that there's a lot of literature that's using AUC curves with presence and kind of background or pseudo-absence data. Okay? I've been focusing on real presence-absence data, but you will see in the literature methods that apply these approaches, AUC values, to presence and pseudo-absence or presence and background data. That's explained in detail, in particular in the paper by Stephen Phillips in 2006 in Ecological Modelling that you've got on your reference list. It's the kind of standard Maxent, the original Maxent paper. But note that if you're doing that, in a practical sense, your target is never going to actually be 1. You're always going to get a value that's a little bit below 1. Quite how far below 1 is difficult to define, and I'd point you to that paper. That's a bit beyond what we're going to cover right now. But I did want to flag that if you're using, say your default AUC curves that you get from the or AUC values that you get from the Maxent software are based on presences and background data, which is a twist. It's exactly the same principles, but it's a twist beyond what I've just spoken about, which is really assuming that you have real presence and absence data. Okay, and let me just, there's just one reference that's actually cited in what I've just spoken about, but that's all written up in numerous places, but the two kind of places that I'm going to refer you to are the 2007 NCEP module that I wrote that I've circulated to everybody. That this stuff is explained in there in as best as I could do, and it's also covered in the Peterson et al. Ecological Niches and Geographic Distributions book. So those would be two sources to go to for revising this and getting into another level of detail.